But I'd like to start out with a couple of questions about your perspective on this work over the length of your career. You've been working in the HIV field for decades. What got you interested in working on this particular disease in the first place?
Well, my training as a PEN was a basic molecular biology, and from the get-go, we knew that in order to eliminate the virus, you need to remove the viral genome, which is incorporated in the host gene. But we didn't have a tool. And then when the gene editing, especially CRISPR technology, was invented in the early 2013, we immediately adapted that technology to HIV. And soon after that, we realized that we were able to take away the virus completely from the T cells and convert the infected T cell become naive, uninfected T cell line. But the challenge for us after that was that can we take this bench work to clinic? In order to do that, we needed to have the technology which can basically deliver the CRISPR-Cas9 to all the infected cells. But the challenge with that approach was that can we identify the infected cells? We decided to utilize the methods of the delivery, which is in this case harmless virus AAV9 to deliver our CRISPR technology to everywhere in the cell, because we really didn't have too much good knowledge, still don't have, that which virus, which cells in the human body are basically hosting the virus genome. So we decided to spread the Cas9 in everywhere, and if there is a virus here in these cells or the other cells, gets in and eliminate. And I think those 30% of animals received basically our therapeutic molecule, and the result was complete eradication of the virus.
So I think this is one of the things that's kind of mind-bending about how we think about this technology, because on one hand, you're using a virus to attack a virus, right? You're using the beneficial aspects of how viruses replicate and infect cells to deliver the cure that will eliminate another virus.
Yes, but as you just said, that's correct. As a virologist, we always try to get the best of the viruses. So in this case, we utilize that the AAV as a tool, as a delivery tool for deliver the CRISPR-Cas9 to all the other cells, which it did. And because still delivery is a big issue in the gene therapy, the approach is gene therapy. And for us to effectively deliver a therapeutic molecule, in this case CRISPR-Cas9, we needed something that universally spread in every cells and tissue, which it did.
Yeah. And so the AAV is a virus that's been modified to be harmless.
That's right. It is adeno-associated virus, which is totally harmless, and it's nothing that 10 years we come and scratch our head. We said, okay, I wish we didn't use AAV because it's harmful. It's not. So as a virologist, we know that this is a completely harmless, and they can deliver the cargo, in this case, CRISPR to every cells.
Right.
And then our cargo, CRISPR, only targets the HIV genome, not the cellular genes. So safety and efficacy was proven already in our laboratory in preparation for clinical trials.
So it really sounds like when this CRISPR technology came along, it was a game changer after all of the work that you've been doing. What surprised you about the results?
Precision and efficacy. That because before that, I remember that when I started my basically studies at Penn, I was working in the laboratory that we were trying to find a way to inactivate the gene in order to understand the function of the cellular gene. But when with the AIDS epidemic, I was at that time I was at the NIH and doing my fellowship in molecular virology, And I thought that can we utilize that thoughts and idea for HIV? But we knew that the efficacy is not there and the specific precision is not there. So you probably can target HIV and 10,000 other cellular genes. So you don't want that. But the CRISPR technology is such a fascinating technique. You could imagine that with 20 nucleotides in size, you can identify incorporate the viral gene in 6 billion nucleotides of the human genome. In less than 24 hours, eliminate the virus completely out of the cells. That is mind-boggling for the scientists. You could imagine that things happen. And then for a lot of supplemental studies that we have done, I didn't show that. We took all those cells and every single nucleotides of the cell was sequenced and verified 100 times to see whether or not the specificity and safety is in place.
Right, so you hear a lot of analogies about CRISPR being like finding a needle in a haystack, but you need to check every other piece of hay in the stack,
very big
right?
Haystack, yes.
To make sure that there aren't any off-target effects.
Yes, that is important. That to make sure that there is a safety, and then so far things seems moving in the right direction. We are very happy with that. One of the things was from the very beginning, we look at the HIV disease not as an infectious disease. That's very important because infectious disease, you try to use an immune cell vaccine and to either immunologically stop the virus growth or eliminate the infected cells. And even better, if you can use to prophylactically develop the basically methods as a vaccination. We look at the disease as a genetic disease because when the virus genome is incorporated in your gene, it's no longer an infectious disease in that person. It's a genetic disease. So if you want to cure that person, you can throw in the body of the person as much vaccine as you can. It's not going to work, as we learned that over the years. But you can eliminate the viral genome and you bring a sterilizing cure to the person, which seems it happened in those experimental animal, and in the monkey. Hopefully, we will get the outcome of that very soon.
So that's a really interesting way
of thinking.
About,
Yes.
But that actually ties into a couple of questions that we have from the audience. Is there a potential for using CRISPR technology or these deactivated virus from the T cells to potentially create a vaccine that addresses the preventative aspect?
Actually, we did. In the very first studies that we did, we performed and published in PNAS, one of the top journals in biomedical field. We demonstrate that the cells, which are virus-free by CRISPR, as long as they have retained that the CRISPR technology, CRISPR molecules in the cells, that cell becomes resistant to reinfection by the virus. It means that every time the virus gets into the cells, as I showed in my third slides, when the RNA becomes a DNA, CRISPR immediately cleaves the viral DNA. So it doesn't even let that integrate into the cells. So prophylactically also has a capacity to prevent reinfection or infection with the virus.
Oh, that's really fascinating. So it could be used.
Yeah, absolutely.
As both.
Yes.
So you had mentioned that in your early experiments that you were seeing a rate of about 30% of eliminating the HIV DNA. What are the factors involved in that success rate, and have you had further success in pushing that rate up?
Yes, actually, what I mentioned, that experiment was initiated about 2 years ago, but it's a very lengthy process, and then the outcome that you see, the paper was published, was the result of it 2 years ago. Right now, we have improved our technique and developed we did some dose escalation, and we know that how much virus or the therapeutic virus we need to put in the animal, and whether or not we should also target some other gene in the cellular gene, which helps the virus to replicate in combination of the targeting HIV. And using that system in collaboration with my colleague, we right now brought up that 30% to 60%, but we are not satisfied. We want to bring it to 100%. So be able to completely eliminate in the monkeys, about the 10 out of 10 animals or whatever number we are using, be cured completely.
So to that point, as you're trying to improve your techniques, there's another audience question asking about how the HIV virus might be changing. We talked about how that mutation rate is very high.
Very good questions. And again, in order for us to target the virus genome, we did bioinformatics studies very carefully in the more than the 1,200 viral isolate from the different patient, which is basically information comes from Los Alamos bank of the HIV. And we identified the region within the virus which never changes. Although the virus changes, as you said, it's true, but the changes happen in the region of the virus, which is for capsid protein, so-called GP120 protein. But we identified the region within the two ends of the virus, which are very conserved, never changes. The second point is that the technology that we have developed, we can easily and quickly, we can personalize the excision strategy that we use, which means that we can utilize that the molecule that we have already in the laboratory in the patient, and the outcome shows that it's not functional. We can take the blood samples from the patient and the sequence that area of the viral genome and see whether or not there is a variation between our technique for our guide RNA and the viral isolate in that person. If we see it's a different, we can immediately create a guide RNA which is basically infecting that individuals and utilize that. That's the personalizing the technique.
Right. So that again.
So actually, we are in process of doing with our colleagues at the Drexel University.
So again, that speaks to the simplicity of the CRISPR technique.
Exactly.
That it's very easy to customize.
Yes.
While at the same time, you are still targeting those parts of the protein that are less likely to change.
Yes.
So, one of the things that I was really impressed that you mentioned was you're developing this therapy with a mind for how it's going to be used in the future and how to make sure, it's accessible to patients who may not have the capacity to reach the medical facilities needed for some of these other strategies for treatment. And as we were talking before we came on stage, we were talking about really the activism of the HIV community, both here in the U.S. and around the world, in really trying to ensure that that equity.
Oh, absolutely.
For all patients.
At the Temple University in the infectious disease department, they have we are very active to reaching out the HIV positive infected individuals, and we are in constant communication with them. And they're actually very supportive to our research, and they reach out themselves and they try to support financially with a limited amount. But it's a good shows that we are working together for the disease which they have been suffering, And
that's such a great example of how
one of the individual that was very interesting. Interaction I had was I was just walking in our department routine, checking out with some of the individuals who are working in this field, and there was a person came and approached me, and he said that I know you, I have seen you, and you're trying to cure the HIV. I said, I'm trying to do my best, but it's not me. It's these are the people, and then we are trying to do. He said, I very much appreciate it. I wish it happened about 5 years ago. I said, why? He said, my brother died of HIV 5 years ago. And see, you're talking earlier to me, what is your motivation for doing this kind of research? That is the motivation. So you're basically trying to help individuals that basically AIDS touch everybody. At the beginning, I remember that we didn't even know the name of the disease. We call it GRID, gay-associated infectious disease, right? So, but then we realized that this is a disease which is caused by HIV, and we learned about the virus, we learned about the antiretroviral treatment. Now we are learning about the complementary technique to eliminate for good virus infection.
So I think an expected question from the audience here is, what does the timeline look like for bringing this type of treatment to clinical trials and ultimately more widespread?
Yes, I mean that the strategy that we used, that the last slide that we showed, that over the last, started actually design an experiment to 2013, and then right now we are at the doorstep for the clinical trial. That for any other the drugs development, if someone is familiar with the system, may take about up to 20 years. We are trying to push this one, so accelerate the process, and then we are in constant communication with the FDA, and they understand that the importance of that technology, and then the promise that this technology gives us in order to cure the disease, and then we will hopefully start the phase 1 clinical trial next year, and maybe we can accelerate the phase 1, phase 2, and based on the outcome of that, hopefully we can get into the clinic for widespread distribution of the drug within the 5 years. That's my hope.
Wow.
But that's something that we are hoping we get there. At maybe very ambitious, basically goal, but when we started in 2014, everybody thought that, how are you going to get into the animals? How are you going to do that one? Okay, so we have a wonderful group of people at Temple, very dedicated, very knowledgeable, and there are a lot of group of scientists that we are working. Some of the friends that right now my friend they were my competitor over last 30 years. We realized that if we want to do something positive at this stage of our career, and we want to basically, why don't you team up and put our expertise together, complement each other, and get rid of this problem? And that exactly happened. Dr. Gendelman was my competitor for 40 years. Actually, we were at NIH at the same time. And then, but his research area is different than mine. But so I'm mostly on the molecular virology. He was mostly on the cell biology. So we combined our effort and the knowledge, and we were able to pull it out.
Such a great story about how science can work.
And needless to mention that he's from Philadelphia, and every time that the news was going up there from my laboratory showing that we were able to eliminate the virus, he was receiving a phone call from his 93 years old mother. Howie, why can't you do these things?
So that's a real reason why I decided to work with you instead of
Actually, she became one of our very good friends, and she comes to Philadelphia. We always go to one of the local restaurant, main line. So we sit down, we talk. It's wonderful, wonderful. It's a great story. I guess we enjoy that.
It's a great partnership.
Yes.
One other question that came up as people are thinking about the future of this treatment, you mentioned that with the antiretroviral therapy, you have to constantly take this medication for the rest of your life. With this treatment, does that solve that problem? And if so, will it be more cost effective?
There won't be any virus to stop it.
Yeah.
Okay. The antiretrovirus therapy has been great. Don't take me wrong. It's a fantastic, converted the disease, deadly disease to chronic illness. But it comes with a baggage. and that is about all those comorbidities that you see. And plus, recently they have found that in Africa there is a lot of virus escapes from the antiretrovirus, basically become resistant to the antiretrovirus, and that is increasing in the really fast paced, and then making the worry. Companies are worried about that. The drug companies are worried about that, and more importantly, patients.
Sure.
They think that okay, today is antiretrovirus, stop my virus. Next day, is it going to do the same or not? Or the new version of the virus is growing in my body. So
Right.
when you eliminate the virus, you don't have to worry about that. It's like a cancer cells, that when the cancer is eliminated, that there's no concern that the cancer is going to come back.
So this is, again, in context of somatic cells. So you're eliminating the gene in an individual, and you're eliminating all of that virus from their cells, but that's not going to be passed down. So I think this is a good opportunity to transition to another instance of where we've been hearing about CRISPR and HIV in the news. So as we said, Dr. Khalili's work is really focusing on somatic cells, but others have been thinking about editing germline cells where those changes are passed down from generation to generation. And you may have seen headlines that came out last year and are still ongoing about a Chinese scientist named He Jiankui, who tried using CRISPR to change CCR5, remember that gene, that receptor gene sitting on the outside of the cell, that was also involved in the Berlin patient. So what this scientist did was actually try to use CRISPR to modify that CCR5 in embryos, so that these twins were then born with modified CCR5 that they will now pass on for generations and generations. And what I thought was interesting about the rationale for this, and this has raised a lot of ethical questions, which I'd like to hear your thoughts on, was that the social stigma around HIV, which exists globally but is particularly stigmatized in China, that the risk that the patients, or rather the parents of these twins who participated in the study, perceived that the stigma of HIV was worse than the potential risks of exposing their children to this new untested therapy. So there's so many ethical questions around this that we can start with, and I'd like to start asking you about your perception of the ethics of just germline gene editing in general, and then we can get a little bit more into the specifics of this case.
I think it's wrong. That's my first response to when I heard about this. I think it's wrong because for multiple reasons. One of the things here is that we're still learning about the CRISPR technology, and then what if that you alter another gene in addition to the CCR5, okay? Then if you do that one, that basically that gene is going to transfer to the generations, okay? And then we're still learning about the CRISPR technology. In our case, we're doing germline. You're trying to basically eliminate the virus from the cell, which is not transferring, that gene is not transferring one cell to another cells. But in this case, it does. And then the regulation and everything in the United States is a very different level of the regulation that we are following, which I'm very happy about that. I'm not sure that we are at the same level in the different environments in maybe China or other place. I think this is very dangerous a strategy, and then the outcome is yet to know. And in fact, we learned that the utilization of this technique by eliminating CCR5 in the germline, Berlin patient was in the germline.
Right, that was another somatic.
Okay, that's a big difference between that and this one. And then elimination of in the germline reduced the lifespan in those the twins already. And then to me, that if you want to do any manipulations or any treatment, you have to get the consent from the patient. You don't have a consent from those. Is a father who happened to be HIV positive, didn't want to have a stigma for these kids.
And also, when we think about using CRISPR for curing diseases, given all of the risks, one of the other guiding principles against germline editing has been to consider whether there are existing treatments for that particular disease. And in this case, whether it's the ART or your treatment that you're developing, it would seem that there are sufficient alternatives.
But you know, I mean, that you can start with the ART to suppress the virus, and then and actually there was a Mississippi baby that was another case of the treatment. When the baby was born with the mother who was HIV positive, immediately put them in the high dose of the antiretrovirus. So the baby was basically virus-free for a couple of years, but unfortunately virus came back and replicates. So at least they could have used some method like that to buy time until the alternative method is developed, which is probably safer, and it's not that drastic. I think CRISPR technology is a terrific, which I think is revolutionizing our life, not just as a disease like a virus or a cancer that you can repair, BRCA1, and then prevent the breast cancer disease or other things. It also can revolutionize the plant industry, and then many other methods. So changing our life to the better rather than just shorter.
So getting back to the bigger question here, I think this particular case study has elicited some other questions about ethics from the audience. Who owns the CRISPR technology? And as we're thinking about regulations, what kind of safeguards do we need to be thinking about to keep drug companies from maybe overcharging? Or
utilization of the CRISPR technology using the double guide RNA, multiplex guide RNA is basically being invented. Temple had the license of that, and right now the company which we mentioned that the license, that the technology, they own it. But for other methods, it's whoever they discovered the Cas9, which is in the Berkeley, and also in the White Institute, Broad Institute in MIT. Yes.
So
this you may need to license those for the utilization of clinic.
So this is still very much an evolving space for regulations.
Yes. It's a very new basically approaches.
So stay tuned. So I want to end thinking about how this technology can be used more broadly. There are questions about, you're using this to treat HIV. What other viruses are we really trying to attack next?
Yes,
and how can this
we have actually developed and published paper on utilization of this technique. Not the same guide RNA, the CRISPR for HSV, herpes simplex virus type 2, and also another virus by the name of JC virus, which causes a neurologic disease, fatal disease, and there's no cure for that. So we are utilizing this technique for other viruses. And then two other viruses, and HPV also, Hep B virus.
So we should expect to see, this application really grow in the next few years.
We are very much focusing on the viruses because of my experience, the education in the human viruses, but the others are using for other techniques.
Well, thank you so much for sharing this really exciting work. Please join me in thanking Dr. Khalili.